Hello friends, I am here with my channel Solving Questions with Gauri ma'am. In continuation with our topic, we are discussing adaptive immunity. We are going further for understanding what is adaptive immunity in detail. See here, we all know we are having bone marrow. So bone marrow is present in long bones, our hand bones, leg bones. Okay, there the bone marrow is present. In the bone marrow, the process of hematopoiesis is taking place. What is this hematopoiesis? Yes, this is a process for the formation of blood. Through the hematopoiesis in the bone marrow, the blood is formed. This process is going on continuously. When we say blood, what are the components of blood? Blood contains plasma, serum, WBCs, RBCs, and it is having platelets. Okay? Plasma and serum are having different proteins. WBCs are there for fighting against antigen. RBCs are there for the carrying of oxygen, and platelets to clot the blood or for blood clotting. Right? Now when we study immune system, we study about WBC. Okay? White blood cells. And all white blood cells are going to form in a bone marrow through the process of hematopoiesis. Yes, moving further with focusing on WBCs. So this is a hematopoiesis, and here lymphocytes are formed and antigen presenting cells are formed. These all cells what we are studying are components of adaptive immunity. Lymphocytes can be a B cells or it can be a T cells, and antigen presenting cells, the examples are dendritic cells and macrophages. Sometimes this B cells can act as a antigen presenting cells. So let's move further and find the detail of each and every cell. Yes, about B cells. So these are synthesized and matures in bone marrow. That's why B cells. Remember this, B cells synthesized and mature in bone marrow. This is a diagram, simple diagram of B cell. It is having nucleus and it is having surface receptors. Membrane bound receptors are there on the B cell, which we call it as a antibody. If we see the structure of antibody, it's a Y-shaped structure. It is having a combination of two heavy chains and two light chains. All are linked with each other with a disulfide bond. It is having N-terminal end and it is having C-terminal end. This is a glycoprotein. Towards the N-terminal end, there is a binding of antigen. So, one B cell, when encounters antigen, can synthesize and express around 2000 antibodies per second. See how vast number is there. So, it can synthesize and express 2000 antibodies per second. Yes, here this is a naive B cell. Naive B cell is the B cell which has not encountered antigen yet. This is a newly formed B cell which is circulating in blood. As soon as it forms, it encounters the antigen, it starts dividing. It proliferates and divides. Okay? It divides into two cells, memory B cell and effector B cell. Effector B cell is also called as a plasma cell. Now this effector B cell secretes enormous number of antibodies. Right? The lifespan for this effector B cell is very short. It lives for few days, and during this lifespan it secretes number of antibodies. This memory B cell, it is having a longer lifespan and it remembers against which antigen immune response has given. Just in our last video actually, we have seen that specificity and memory, these two are the attributes of adaptive immunity. So here we can see that the B cell, it can have a specific type of antibody for different antigens, and again simultaneously it is preparing memory B cell for memorizing the immune response. Moving further to T cells. See here, in the bone marrow through hematopoiesis, T cells are formed. But these T cells get matured in thymus. Okay, we have to remember this, and due to this T, the cells are known as a T cell, right? Thymus is a gland which is present above the heart, and in that these T cells get matured. These T cells get trained. Their training takes place in a thymus, and that's why these are T cells. Now, the T cells are of two types, that is T helper cell and T cytotoxic cells. So TH and TC. Now when we say immune response of the T cells, how it happens? So likewise, the B cells can recognize antigen directly, isn't it? B cells circulate in a blood, and if antigen encounters, then B cells start secreting antibody against it. But in case of T cells, this doesn't happen. It needs presentation of the antigen. We have seen two types of T cells, T helper cell and T cytotoxic cell. So T helper cells are having TCR on them, that is T cell receptors. Along with this, membrane molecules are there known as CD4, cluster of differentiation 4. Now, in case of T cytotoxic cell, it is also having TCR on it, T cell receptor, and along with it, it is having CD8 molecules, cluster of differentiation 8. What is this TCR and CD8? Now try to understand in this way. As I have told you, T cell needs presentation. The antigen has to be presented to T cell. It cannot recognize antigen by its own. So who will do this function? This function is done by APC, that is antigen presenting cell. The name itself is telling us that these cells are going to present the antigens. So this is a combination which we have to remember. If T helper cell is there, it is having CD4 molecule on it. Here it is. This will recognize the MHC class 2 molecule which is present on APC. Okay. Now this APC are antigen presenting cells. So the examples are dendritic cell, macrophages. Sometimes B cell also functions as an antigen presenting cells. What do these APC does? APC does phagocytosis. They circulate in a blood. If they encounter the antigen, they phagocytose that antigen and they present the part of that antigen on MHC molecule which is present on their surface. So due to this presentation, there is a specific recognition. What we have written here. This is a combination to be remembered. If APC is having MHC class 2 molecule, it is getting recognized by CD4 molecule present on T helper cell. Secondly, if APC is having class 1 molecule, it is going to recognize by CD8 present on T cytotoxic cells. Okay. So after phagocytosis, APC is going to present the antigen whether on MHC class 1 or MHC class 2. And accordingly, T helper cell or T cytotoxic cell will recognize it. Antigen presenting cell are the component of innate immunity, right? We have seen in our lectures that phagocytosis is the barrier of innate immunity. So both immunity works hand in hand. It doesn't happen that if adaptive immunity has taken care, then innate immunity sleeps for a time. It also works and this also works. Now, these MHC are the major histocompatibility complex. These are protein molecules present on an antigen presenting cell. Out of this, MHC class 1 is present on all nucleated cells. All cells of body are having MHC class 1 on them, which can present antigen to T cytotoxic cell. Okay. Secondly, this MHC class 2 is present on a phagocytic cell, or we can say antigen presenting cells. This presents the antigen to T helper cell and immune response takes place. Yes, moving further to understand the effects or the action of T cells. See here, when T helper cell get activated after the presentation of antigen with the class 2 MHC on the antigen presenting cell. This gets divided into memory T cell and effector T cell. Now this effector T cell secretes cytokines. These are some proteins which goes and activate other cells of immune system. It activates B cells, T cytotoxic cells. It activates macrophages, and also some other cells of immune system get activated using these cytokines. So T helper cells are very much important as far as immune system is concerned. It is activating all the cells of immune system. And also it is having memory cells. Okay. Secondly, T cytotoxic cell, how it works. So again, it has to be, it needs representation of antigen to itself. This is done by class 1 MHC, which are present on a APC. Now, after the presentation, the T cytotoxic cell proliferates and differentiates. It forms a cytotoxic T lymphocyte. We call it as CTL, cytotoxic T lymphocyte. This itself carries the cytotoxic activity or killing activity. It goes and kills that particular cell. The T cytotoxic cell is active against the tumor cells or the virally infected cells. So, as we can imagine, the tumor cell or virally infected cell has to be killed. There is no option for that. Whether it is a own cell also, but once it is virally infected, we need to kill that cell. So that is done by the T cytotoxic cell. So here we have concluded for today's video. If you like my videos, please don't forget to like, share and subscribe for my channel, and also to hit the bell icon. Thanks for watching.